Understanding the human body is, in many ways, an investment in your own well-being. The more familiar you are with how your body works, the more likely you are to make choices that support your health, recognize early signs of illness, and respond appropriately. This knowledge becomes particularly useful when trying to make sense of the steady stream of information about nutrition, pharmaceuticals, medical technology, and the ever-shifting world of infectious or genetic diseases. Eventually, of course, no one escapes entirely unscathed. At some point, something will go wrong. And when it does, having some insight into your own physiology can make you a more competent parent, partner, friend, colleague, or caregiver. Now, the human body is not a simple machine. It's a complex structure composed of cells, which group into tissues, which in turn form organs, which are organized into organ systems. For the body to function properly, these components must not only operate effectively in their own right, but also coordinate with one another. Many organs wear more than one hat, contributing to multiple systems depending on their unique abilities. Most people are more familiar with the organ level of biological organization, since this is where so many of our everyday health experiences take place. You might have a stomachache, break a bone, or hear about someone battling lung disease or skin cancer. Each of these conditions is associated with a particular organ, highlighting just how much our sense of health and illness is tied to these structures. An organ, fundamentally, is a structure made from different types of tissue working together to perform a particular job that serves the body as a whole. When several organs join forces to perform related tasks, they form an organ system. These systems carry out broader physiological roles. For instance, there's the digestive system, composed of the stomach, intestines, liver, and others, all coordinating to process food and extract nutrients. Though we won't trouble ourselves with tables, you should know that each system includes specific organs tailored to its particular functions. In this discussion, we've grouped these systems into three overarching categories: those involved in control, those dedicated to cell maintenance, and those providing support. These aren't rigid classifications, more like a helpful filing system for your brain. Take the reproductive system, for example. It has been slotted under control because it governs the complex choreography of reproduction. But it's also a maintenance system, producing and managing the very cells required for that process. The takeaway here is not to get too hung up on which box an organ or system is placed in. In reality, just as organs collaborate within systems, systems interact constantly with one another. Every aspect of the body's design, from the tiniest molecule to the largest organ system, is interconnected. We often study these parts in isolation, looking at cells under a microscope or examining the function of a particular organ, but none of them operates in a vacuum. Consider diabetes. This endocrine disorder alters blood glucose levels, but the consequences ripple outward. Wound healing slows down as the immune system struggles. The kidneys, part of the urinary system, may be damaged. The cardiovascular system can suffer, sometimes leading to vision loss due to damage in the blood vessels of the eyes. The body does not compartmentalize the way our textbooks or lectures might. Even the simple act of assigning an organ to a specific system can feel slightly artificial. Most organs moonlight in more than one role. The liver, for instance, doesn't confine itself to digestion alone. It has endocrine and detoxifying duties as well. So, while we use these systems and categories to navigate the landscape of human anatomy and physiology, it's wise to remember, in the end, everything is connected. Each organ system carries out specific responsibilities, and for the sake of study, they're typically examined one at a time. But in real life, they do not operate in isolation. Rather, they form a sort of biological alliance, coordinating their actions to help the body maintain homeostasis, a stable internal environment despite external fluctuations. Consider water balance. The cardiovascular, urinary, and lymphatic systems all lend a hand in this ongoing negotiation. The cardiovascular and lymphatic systems are responsible for transporting fluids and are attuned to changes in solid and water levels, regulating pressure accordingly. If your water levels climb too high, the urinary system compensates by producing more dilute urine, expelling the excess. If levels drop, it does the opposite. Urine becomes more concentrated, conserving what little water remains. Temperature control is another example of collaborative regulation. The cardiovascular, integumentary, respiratory, and muscular systems all participate in keeping your body temperature within a narrow, acceptable range. When you overheat, blood vessels near the skin surface widen, promoting heat loss. The skin begins to sweat, and as that sweat evaporates, it cools the body. Rapid breathing also assists by releasing excess heat. This explains why, during vigorous exercise, you sweat, breathe heavily, and develop a distinctly red hue, though the heavy breathing does double duty, also supplying oxygen to your hardworking muscles and clearing out their carbon dioxide. If the temperature drops too far, the response flips. Blood vessels in the skin constrict to preserve core heat, reducing blood flow to your arms and legs. Muscles shiver, contracting and relaxing quickly to generate heat. The fine hairs on your skin stand up, theoretically trapping a layer of insulating air, though in humans, this is more symbolic than effective. These responses account for the chills, goosebumps, and numb fingers that come with cold weather. Calcium regulation is another tightly monitored function, with the endocrine system overseeing the operation. Calcium is required for an impressive list of tasks: keeping the heart beating, enabling muscle contractions, activating enzymes, and facilitating communication between cells. We obtain calcium from our diet, and the small intestine handles its absorption. Here, the parathyroid and thyroid glands step into their supervisory roles. When calcium in the blood dips too low, the parathyroid gland releases parathyroid hormone. This hormone instructs the kidneys and bones to retain or release calcium accordingly. The kidneys stop excreting it in urine, and osteoclasts in bone tissue break down bone to liberate more calcium. When blood calcium rises too high, the thyroid responds with a hormone of its own, calcitonin. This hormone tells the kidneys to release more calcium in the urine and slows the production of active vitamin D, reducing calcium absorption in the gut. Meanwhile, osteoblasts in the bones take the excess calcium and put it to use in building bone. These responses, all orchestrated by endocrine signaling, help restore balance. The regulatory story continues with blood glucose levels. The pancreas and liver work in tandem to maintain this particular equilibrium. If blood glucose drops, pancreatic cells release glucagon, which prompts the liver to break down glycogen into glucose, a process called glycogenolysis. The glucose then re-enters circulation, available for cellular energy. When glucose levels are too high, the pancreas produces insulin instead. Insulin encourages the conversion of glucose into glycogen, a process known as glycogenesis, which the liver then stores for future use. Even your hunger pangs play a role. When blood glucose is low, the nervous and digestive systems conspire to make you uncomfortably aware of it. Your stomach may grumble, and you might feel an insistent emptiness. These signals prompt you to eat, correcting the glucose deficit. In all of these examples, water regulation, temperature control, calcium balance, and blood glucose levels, multiple systems cooperate to maintain internal stability. The body is, if nothing else, a master of teamwork. The central nervous system, or CNS, is composed of the brain and spinal cord, both of which are protected rather carefully by bone, membranes, and fluid. The brain, housed snugly within the cranial cavity, includes three major parts: the cerebrum, cerebellum, and brainstem. The nervous system also involves cranial and spinal nerves that serve as its primary communication channels. This system serves three main purposes. It collects sensory input, integrates that information, and then sends out motor commands. Sensory input is gathered by neurons and supported by glial cells and synapses. Neurons, the excitable workhorses of the system, transmit impulses from sensory receptors to the CNS. Once there, the brain processes the data, a function reserved exclusively for this cerebral hub. After integration, motor output occurs as the brain and spinal cord dispatch impulses to muscles and glands. While the CNS, composed of the brain and spinal cord, acts as the control center, it works closely with the peripheral nervous system, or PNS. The PNS consists of a sprawling network of cranial and spinal nerves that connect the body to the CNS. Within the PNS, sensory receptors detect changes in the internal and external environments and transmit this information to the CNS via afferent nerves. The PNS then carries out responses through efferent nerves. The PNS is further divided into the autonomic system, which governs involuntary functions such as heartbeat and digestion, and the somatic system, which manages voluntary control of skeletal muscles, skin, bones, and joints. These two systems interact continuously, sharing nerve pathways and maintaining a cooperative relationship. The CNS is not merely a switchboard for input and output. It processes information and, according to a number of studies, may generate motor activity independently of sensory input. In other words, it behaves rather like an autonomous system. Perhaps something to ponder the next time you move before thinking. Damage to the CNS or entrapment of peripheral nerves can produce a wide range of dysfunctions. These range from altered facial expressions, such as a lopsided smile, to more systemic issues like impaired lung or bladder function, weakened limbs, or even changes in sexual response. Headaches, sinus problems, and sore throats may also be linked to such neural disruption. Even when muscles are used regularly, they can weaken or atrophy if the nerves connecting them are impaired. The structure of neurons reflects their function in transmitting and processing information. Neurons vary widely in shape, size, and electrochemical properties, but all share a few common features. The soma, or cell body, contains the nucleus and handles protein synthesis. It ranges from 4 to 100 micrometers in diameter. Dendrites extend from the soma like branches from a tree, forming what's aptly called a dendritic tree. These are the primary sites for receiving input, though under certain conditions they can also send signals. The axon is a thin, cable-like projection that carries impulses away from the soma. Some axons extend to extraordinary lengths, over a meter in humans, from spine to toe, and several meters in giraffes. At its origin, The axon emerges from the soma at a region called the axon hillock. This area has a high concentration of sodium channels, making it the most easily excitable part of the neuron and the likely site where nerve impulses begin. Though typically the channel for outgoing signals, this area can also receive input from other neurons. The axon ends at the axon terminal, a specialized structure designed to release neurotransmitters and communicate with other cells. Though neurons are traditionally depicted with well-defined roles, dendrites for input, axons for output, reality is more flexible, and the actual behavior of these components is often more complex. In the central nervous system, axons and dendrites are usually about a micrometer thick. The soma typically measures between 10 and 25 micrometers, not much larger than the nucleus it houses. The longest human sensory neurons run from the toes to the dorsal columns of the spinal cord, a distance of over 1.5 meters. Much of what we understand about axonal function has been gleaned from the giant axon of the squid, prized in research for its impressive size, 0.5 millimeter thick and several centimeters long. Functionally, neurons fall into several categories. Sensory, afferent neurons bring information into the central nervous system from tissues and organs. Motor, efferent neurons carry signals out to effector cells. Interneurons connect neurons within regions of the CNS. The terms afferent and efferent can also describe the direction of information flow relative to brain regions. Neurons can also be classified by the nature of their influence. Excitatory neurons activate their target cells. These include most motor and somatic neurons, and often use glutamate or acetylcholine as their neurotransmitters. Inhibitory neurons suppress activity in their targets. These are also known as interneurons or microneurons and are found in structures like the neostriatum, globus pallidus, and cerebellum. They typically use GABA or glycine. Modulatory neurons don't simply excite or inhibit. They fine-tune responses through neuromodulation. These neurons use substances like dopamine, serotonin, and acetylcholine to produce subtler, more complex effects. Each synapse, a junction between two neurons, can receive both excitatory and inhibitory signals. The result depends on summation, a mathematical balancing act that determines whether the neuron will fire. The endocrine system is a control network composed of ductless glands that release hormones into the bloodstream. These hormones serve as chemical messengers, traveling through the circulatory system to distant cells, which interpret the signals and act accordingly. At first glance, it may seem improbable that a tiny chemical could circulate in the blood and trigger a specific response far from its origin. Yet this occurs continuously and quite reliably in our bodies. Hormones are responsible for maintaining homeostasis and coordinating responses to internal and external stimuli. Without them, basic life functions, growth, temperature regulation, reproduction, even metabolism, would be impossible. The endocrine system establishes an electrochemical connection from the hypothalamus in the brain to virtually all organs that govern metabolism, development, and reproduction. It secretes two main types of hormones: steroidal, lipid-based, and non-steroidal, protein-based. Regulation of hormone levels is typically managed through negative feedback mechanisms. When hormone levels rise, the system responds by dialing back production. There are exceptions to this pattern, childbirth being a notable one, but most hormonal activity is governed by this stabilizing loop. Other influences, including immune responses, also help regulate hormone levels. It's useful here to distinguish between endocrine and exocrine glands. Exocrine glands, such as sweat, salivary, mammary, and pancreatic glands, use ducts to deliver their secretions either to the outside of the body or into the lumen of an organ. These are not part of the endocrine system. In contrast, endocrine glands lack ducts entirely. Instead, they release their secretions directly into intercellular fluid or into the bloodstream. The major players in the endocrine system include the pituitary gland, thyroid gland, parathyroid glands, adrenal glands, pancreas, and gonads. The pituitary gland, often dubbed the master gland, is attached to the hypothalamus in the lower forebrain. The thyroid, consisting of two lobes connected by a narrow bridge, sits just below the larynx and is affixed to the trachea. The parathyroid glands, 4 in total, are embedded behind the thyroid lobes. Each adrenal gland rests atop a kidney, with a distinct outer cortex and inner medulla. The pancreas is located along the curve of the stomach, near the first section of the small intestine, the duodenum. And the gonads, ovaries in females and testes in males, reside in the pelvic cavity. A hormone, simply put, is a chemical signal. It enables one cell to influence the behavior of another, often at considerable distance. These hormones, produced by the endocrine glands, enter the extracellular space and then diffuse into capillaries, hitching a ride through the bloodstream to their targets. Their job is to modulate activity in specific cells, called target cells, and thereby maintain internal balance, regulate growth, and oversee development and reproduction. While the endocrine and nervous systems both aim to coordinate the body's responses, their methods diverge. The nervous system uses neurotransmitters to produce almost instantaneous effects on adjacent cells, be they muscle, gland, or other neurons. These effects, though fast, are fleeting. Hormones, in contrast, take longer to act, ranging from seconds to days, and their influence is more widespread. They can affect any number of cells, near or far, and their effects persist as long as the hormones remain in circulation, which may be for hours. Reproduction, simply put, is how living organisms make more of themselves. It's the defining difference between the living and the non-living, though, ironically, it's not required for an individual to survive, only the species. In the human version of this age-old biological ritual, 2 sex cells are involved. The sperm, contributed by the male, and the secondary oocyte, contributed by the female. Only after fertilization does that secondary oocyte officially earn the title of ovum or egg, a technical detail often lost in casual conversation. To produce new life, both the male and female reproductive systems are required. Each is specialized to produce, nourish, and transport gametes, though their shapes and arrangements differ significantly. The male's reproductive organs span both inside and outside the pelvic cavity, while the females are housed entirely within. The male system includes the testes, where sperm are produced, and a series of ducts and glands that transport and support them. Sperm travel through the epididymis, vas deferens, ejaculatory duct, and ultimately the urethra. Along the way, several glands contribute secretions that make up semen, the transport medium for sperm. These glands include the seminal vesicles, the prostate gland, and the bulbourethral glands. The seminal vesicles, located near the base of the bladder, contribute the majority of seminal fluid, around 65 to 75%. This fluid includes nutrients like fructose, along with enzymes, mucus, and various signaling molecules to support and energize the sperm. The prostate gland, encircling the urethra just below the bladder, contributes a clear, slightly alkaline fluid that helps neutralize the vaginal environment, accounting for about 25 to 30% of semen. The bulbourethral glands, small pea-sized structures situated behind the prostate, secrete a gelatinous fluid, pre-ejaculate, which lubricates the urethra and flushes out any lingering acidic urine or foreign material. Though small in volume, their contribution is not without purpose. The epididymis is a tightly coiled duct lying just outside each testis. Here, sperm are stored and matured until they're summoned for use. The vas deferens, roughly 30 centimeters in length, transports mature sperm during ejaculation, delivering them to the urethra while picking up secretions from the accessory glands along the way. The urethra itself serves a dual purpose in males, conveying both urine and semen to the outside world. And then there's the penis, made of erectile tissue, 2 corpora cavernosa and 1 corpus spongiosum, with the urethra passing through. It's the final delivery mechanism, both for reproduction and urination. At the core of it all are the testes, housed in the scrotum outside the body. These are the male gonads, responsible not only for producing sperm, but also for synthesizing testosterone, thanks to the activity of Leydig cells. When sperm meets secondary oocyte, fertilization can occur. This usually takes place in the uterine tubes, also known as the fallopian tubes, which stretch from each side of the upper pelvic cavity. Here, the gametes unite to form a new individual, and if all proceeds as nature intends, the female then carries the resulting embryo through pregnancy and birth. Though the machinery differs, the goal is shared: continuation of the species through the merging of 2 gametes into one new life. A straightforward concept, executed with a remarkable amount of biological choreography. The female reproductive system serves several essential functions. It produces gametes in the form of oocytes, or eggs, secretes sex hormones, receives sperm during intercourse, protects and nourishes a fertilized egg throughout pregnancy, delivers the fetus during childbirth, and, following birth, provides nourishment to the newborn via milk from the mammary glands. The ovaries are the primary reproductive organs, or gonads, of the female. Located on either side of the uterus within the pelvic cavity, these ovoid structures house ovarian follicles. Each follicle contains an oocyte, which may be released during ovulation as part of the menstrual cycle. The fallopian tubes extend from the sides of the uterus and reach toward the ovaries, although not physically connected. These tubes transport the oocyte toward the uterus and serve as the typical site of fertilization. The uterus itself is a muscular pear-shaped organ divided into 2 main parts, the upper fundus and the lower cervix. It is the location where fetal development takes place during gestation. Below the uterus lies the vagina, a muscular canal lined with mucous membrane. It functions as the passage for menstrual discharge in childbirth, and also receives sperm during intercourse. Externally, the vulva includes structures such as the labia majora and minora, mons pubis, clitoris, and vestibule, along with associated glands. These parts are heavily innervated and contribute to sexual arousal and pleasure. The perineum, the area between the vaginal opening and the anus, supports the pelvic floor muscles and can be affected during childbirth. The mammary glands, located over the pectoral muscles, are not technically part of the reproductive tract, but they do play a key role in reproduction by nourishing the infant through milk production. Although male and female reproductive systems have distinct roles, they share foundational similarities. Both develop from homologous embryonic tissue, possess gonads that produce gametes, and undergo changes during puberty triggered by hormonal signals. The primary functional difference lies in the nature of gamete production. Males produce sperm continuously after puberty. In contrast, females are born with a finite number of gametes. During fetal development, the female's ovaries form 6 to 7 million oogonia by the 5th month of gestation. These cells begin meiosis, becoming primary oocytes, and then pause in prophase 1. This arrest continues from birth until puberty. At puberty, the menstrual cycle begins. Each month, typically one oocyte resumes meiosis, completing the first division to form a secondary oocyte and a polar body. The process then pauses again, this time in metaphase 2. If fertilization occurs, meiosis is completed, resulting in a mature ovum and another polar body. A newborn female has about 1 million oocytes, but by puberty, this number drops to between 400,000 and 500,000. Over the course of her reproductive life, only about 500 to 1,000 of these will actually be ovulated. The rest degenerate over time. With age, both the number and quality of oocytes decline, continuing until menopause, typically occurring around age 50, at which point ovulation and menstruation cease entirely. The oocytes present at birth are all a woman will ever have. No new ones are made. They age as she does. Our experience of reality depends entirely on our senses. These physiological channels allow us to detect, interpret, and respond to the stimuli around and within us. Though commonly boiled down to the familiar 5, sight, hearing, touch, taste, and smell, our sensory system is more complex. Depending on how you define a sense, humans have at least 7, possibly more. Neurologists continue to debate the precise number, partly due to the blurred boundaries between sensory modalities. These senses are typically divided into 2 categories: exteroceptors, which perceive stimuli from the external environment, such as smell, taste, and balance, and interoceptors, which monitor internal conditions like blood pressure, glucose levels, and pH. Individual sensory experience is subjective. Taste, for instance, can vary significantly from person to person. What delights one palate might repulse another, because the brain interprets incoming stimuli uniquely based on a range of biological and experiential factors. Taste, or gustation, and smell, or olfaction, both fall under the broader category of chemoreception. These senses rely on specialized receptor cells that respond to specific chemical compounds. When a chemical interacts with these receptors, a signal is sent to the brain, which then identifies it as a particular flavor or odor. Taste is sensed by taste buds, which are distributed not only across the tongue, but also in areas such as the soft palate and epiglottis. These taste signals travel via 3 cranial nerves. The facial nerve, cranial nerve 7, handles taste from the front 2/3 of the tongue. The glossopharyngeal nerve, cranial nerve 9, handles the rear third, including the circumvallate papillae, and the vagus nerve, cranial nerve 10, picks up taste from the throat and epiglottis. All taste buds are capable of detecting all taste types, though some areas may be slightly more sensitive to certain tastes. Taste buds reside on structures called papillae, which come in 4 varieties. Fungiform papillae are mushroom-shaped and found mostly at the tip of the tongue. Filiform papillae are long, thin, and mechanical in function. They do not contain taste buds. Foliate papillae appear as ridges and grooves toward the rear sides of the tongue. Circumvallate papillae, fewer in number, form a circular row near the back of the tongue, just in front of the sulcus terminalis. Olfaction, the sense of smell, operates via a similar principle. Airborne molecules dissolve on the moist epithelium of the nasal passage, triggering olfactory receptors. These receptors send impulses to the brain through the olfactory nerve, cranial nerve 1. Remarkably, up to 90% of what we perceive as taste is actually derived from smell, which explains the dull flavor of food during a head cold. Genetically, humans have around 347 functional odor receptor genes, though this number varies among individuals and populations due to gene mutations. One common example of olfactory variation is the inability of some people to detect androstenone, a molecule found in male sweat. For those who can smell it, the scent ranges from faintly sweet to unpleasantly musky. Yet another reminder that sensory perception is as much personal as it is physiological. Each olfactory receptor neuron in the nose is uniquely specialized, expressing just one functional odor receptor. These neurons operate much like a key lock mechanism. If an odor molecule fits the receptor's configuration, the neuron responds. One interpretation of this mechanism is shape theory, which proposes that each receptor is tuned to detect a specific feature of an odor molecule. A variation on this is the weak shape or odotope theory, suggesting that each receptor recognizes only fragments of molecules. These minimal signals are then combined to create a more complex perception, much like how the visual system assembles basic inputs into a detailed image. Another more controversial model, vibration theory, posits that receptors detect the vibrational frequency of molecules through a mechanism involving electron tunneling. While interesting, its behavioral predictions haven't held up well under scrutiny. Olfactory receptor neurons, also known as olfactory sensory neurons, are the primary cells responsible for translating chemical stimuli into neural signals. In humans, there are about 40 million of these neurons, all located on the olfactory epithelium inside the nasal cavity. Structurally, they are bipolar neurons, with dendrites extending into the nasal cavity and axons that pass into the brain through the olfactory nerve to reach the olfactory bulb. Extending from the dendrites are numerous tiny cilia that project into the mucus layer covering the nasal epithelium. These cilia contain the actual olfactory receptors, which are a type of G protein-coupled receptor. Each receptor neuron expresses only one type of olfactory receptor, but the olfactory system compensates by clustering neurons of the same receptor type. Their axons converge to form glomeruli, spherical structures in the olfactory bulb. When an odor molecule binds to a receptor on these cilia, it activates the G protein Golf, which in turn activates adenylate cyclase. This enzyme increases the level of cyclic AMP, which opens ion channels in the cell membrane. As sodium and calcium ions flow into the neuron, the cell depolarizes, generating an action potential. The signal is then passed along to the brain. These receptor neurons don't last forever. They're replaced roughly every 40 days by neural stem cells in the olfactory epithelium. This continuous regeneration represents one of the few known examples of adult neurogenesis in the central nervous system, making the olfactory system a focal point for research into neural regeneration and development. Axons from all neurons expressing the same receptor type converge in the olfactory bulb and synapse with mitral cells. These mitral cells relay the information to higher regions of the brain, where the features of the odor are integrated into a coherent perception. Because most odor molecules contain multiple features, the brain can recognize a vast array of smells from relatively few receptor types. The olfactory system is strongly linked to memory, particularly emotional memory. This is due to its anatomical proximity to the limbic system and the hippocampus, regions of the brain involved in emotion and spatial memory, respectively. This relationship helps explain why certain smells can so powerfully evoke past experiences. Some pheromones are processed by the olfactory system, although in many animals, pheromones are detected by a separate structure known as the vomeronasal organ. This organ lies between the nose and mouth, in the vomer bone. Snakes famously use their tongues to sample molecules and deliver them to this organ. Some mammals display the flamen response, curling back their lips to draw air into it. In humans, the existence or function of this organ remains a subject of debate. Our experience of flavor involves more than taste alone. Taste, smell, and trigeminal sensations, which detect temperature, texture, and spiciness, work together to create what we know as flavor. Human taste is limited to 5 basic types: sweet, salty, sour, bitter, and umami. However, the 10,000 or so scents we associate with food flavors are mostly olfactory. This explains why food tastes bland when your nose is congested. Olfactory function plays a central role in appetite and nutrition. About 80 to 90% of what we think of as taste is actually mediated by smell. As people age, their sense of smell often diminishes, which can negatively affect appetite and, by extension, nutritional health. For the elderly, this decline in olfactory sensitivity warrants attention, especially when monitoring food intake and overall well-being. Vision is a collaborative effort between the eyes and the brain. While the eyes are responsible for capturing the majority of visual stimuli, the brain, specifically the cerebral cortex, processes at least a third of the incoming visual information. It's a partnership that transforms light into meaning. The human eye is roughly an inch in diameter and shaped like a slightly elongated sphere. It resides in a protective bony socket of the skull. Structurally, the eye has 3 concentric layers: the sclera, the choroid, and the retina. The outermost layer is the sclera, a tough fibrous tissue that gives the eye its shape and provides protection. Its visible part is the white of the eye. At the front, the sclera becomes transparent, forming the cornea. The cornea serves as the eye's outermost lens, bending incoming light to help focus it. Beneath the sclera lies the choroid, a thin vascular layer that supplies oxygen and nutrients to the retina. It contains pigmented cells that prevent light from scattering within the eye, which would otherwise blur vision. Light passes through an opening in the choroid called the pupil, whose size is regulated by the iris. The iris adjusts the pupil's diameter based on ambient light. It contracts in bright conditions, making the pupil smaller, and dilates in dim conditions, making the pupil larger. Immediately behind the pupil is the lens, composed largely of proteins called crystallins. The lens adjusts its shape, thanks to the ciliary muscles in the ciliary body, to focus light on the retina, a process known as accommodation. The lens is held in place by fine ligaments called zonules. Together, the choroid, ciliary body, and iris make up the uvea, a term likely inspired by its dark, wrinkled, grape-like appearance when dissected. The visual system must also cope with the challenge of motion. The brain cannot process images clearly if they drift across the retina too quickly. To compensate, it adjusts the eye's position in response to head movement, ensuring that the image of interest remains centered on the fovea, the most sensitive part of the retina. These eye movements are vital for visual clarity. Failure to coordinate them accurately can result in impaired vision. With two eyes, coordination becomes even more critical. Both eyes must aim precisely so that the same object projects onto corresponding spots on each retina. If alignment is off, double vision results. Eye movement, like movement in other parts of the body, is controlled by striated muscles. However, the eyes have their own set of advantages. Their muscular movements are faster, more refined, and more specialized than those of skeletal muscles elsewhere in the body. Another critical aspect of vision is depth perception, the ability to see the world in 3 dimensions and to judge distances accurately. Though commonly conflated with binocular vision or stereopsis, depth perception draws on both binocular and monocular cues. Binocular vision provides a strong foundation for depth perception, but monocular cues, such as perspective, shading, and motion, also contribute significantly. The ear serves as both a detector of sound and a regulator of balance and spatial orientation. It accomplishes these functions through mechanoreceptors, specialized hair cells with stereocilia that respond to physical movement. These sensory cells reside in the inner ear and are sensitive to even minute mechanical stimulation. The ear is divided into 3 main regions: the outer ear, the middle ear, and the inner ear. The outer ear includes the pinna, also called the auricle, the ear canal, and the superficial layer of the tympanic membrane, or eardrum. The pinna, while structurally distinctive, is not essential for hearing, though its folds and curves help gather sound. More critical is the ear canal, a tunnel lined with skin that adheres to cartilage in its outer portion and bone in its deeper regions. Glands in this skin produce cerumen, or earwax, which along with tiny hairs helps trap debris. The outer ear ends at the tympanic membrane, which vibrates in response to sound waves. The middle ear begins just behind the eardrum and houses 3 small bones, the ossicles, known as the malleus, hammer, incus, anvil, and stapes, stirrup. These bones form a chain that transmits vibrations from the eardrum to the oval window of the inner ear. The malleus is attached to the tympanic membrane and passes movement to the incus, which in turn moves the stapes. The stapes, the smallest bone in the human body, presses against the oval window, setting fluid in the inner ear into motion. This chain reaction is finely tuned and essential for converting airborne vibrations into a form the inner ear can interpret. Also within the middle ear is the Eustachian tube, which connects to the back of the pharynx. This tube helps equalize air pressure between the middle ear and the outside environment. The middle ear cavity, similar to the paranasal sinuses, is lined with mucosa and ventilated through the nose. Air spaces in the mastoid portion of the temporal bone also connect to the middle ear, forming a ventilation system for pressure regulation. The inner ear contains the cochlea, the organ responsible for hearing, and the vestibular apparatus, which regulates balance. These structures are encased in the densest bone in the body. The cochlea contains 3 fluid-filled chambers: the tympanic canal, vestibular canal, and cochlear duct, or middle canal. When the stapes moves against the oval window, it creates fluid waves inside the cochlea. These waves stimulate hair cells in the organ of Corti, which is embedded within the cochlear duct. Sound is processed based on frequency. High-frequency sounds strike the basilar membrane near the base of the cochlea, close to the oval window. Lower-frequency sounds travel farther, activating hair cells closer to the apex. The strength of stimulation depends on the volume of the sound, and the resulting neural signals are sent to the brain via the auditory nerve, a branch of the 8th cranial nerve. These signals are first processed in the spiral ganglion, a cluster of nerve cells in the cochlea. Humans can typically hear sounds ranging from 20 Hz to 20,000 Hz. However, sensitivity to high-frequency sounds declines with age, a fact not lost on teenagers who have been known to use high-pitched ringtones above 17,000 Hz to evade adult detection. In addition to hearing, the inner ear manages balance through the vestibular system, which includes the 3 semicircular canals and the vestibule. These structures detect head motion and orientation relative to gravity. The fluid movement inside them bends hair cells, triggering nerve impulses that help the brain determine body position and motion. Hair cells are tall, column-like cells crowned with a bundle of about 100 to 200 cilia, and these cilia are central to the process of hearing. Lightly resting against the longest cilia is the tectorial membrane, which oscillates with sound vibrations. As this membrane moves, it tilts the cilia, opening ion channels and allowing current to enter the hair cell. These cells respond not with the all or none spikes seen in most neurons, but with graded potentials, meaning their response varies with the strength of the stimulus. These hair cells reside in the organ of Corti, situated on a flexible membrane inside the cochlea. When sound waves travel through the ear and reach the cochlea, they cause the oval window to vibrate, pushing fluid through the cochlear chambers. This movement disturbs the basilar membrane, where the organ of Corti sits, leading the outer hair cells to rub against the tectorial membrane. These outer hair cells exhibit muscle-like behavior, amplifying the vibrations, though only the quiet ones. Louder sounds bypass this amplification. The inner hair cells, once activated, transmit signals to the cochlear nerve. The cilia of these cells are connected by tip links, which stretch and compress in response to movement. This mechanical action opens ion channels, generating the receptor potential. The cochlea contains far fewer hair cells than it does afferent nerve fibers. The cochlear nerve, which carries sound information to the brain, joins with the vestibular nerve to form the 8th cranial nerve. At the synapse, glutamate serves as the neurotransmitter. A notable feature of the synapse is the presynaptic dense body, or ribbon, which is surrounded by synaptic vesicles and likely supports the rapid release of neurotransmitter. Efferent signals from the brain also reach the cochlea, connecting to outer hair cells and to afferent dendrites under inner hair cells, playing a role in fine-tuning auditory perception. In the brain, motion detection in sound is handled by the right posterior superior temporal gyrus. The superior temporal gyrus also contains the primary auditory cortex, areas 41 and 42, which handles the raw features of sound like pitch and rhythm. More advanced processing occurs in Wernicke's area, area 22, which distinguishes speech from noise or music and interprets it meaningfully. Further integration of sensory information takes place in the gnostic area of the cerebrum, areas 5, 7, 39, and 40, where multiple sensory inputs are synthesized into a unified perception. Underwater hearing is altered due to the faster speed of sound in water. Sound is conducted mainly through bone, and localization, determining where the sound is coming from, is diminished. In air, sound localization depends on interaural differences, the difference in volume and timing of a sound as it reaches each ear. The brain uses these tiny discrepancies, on the order of 10 milliseconds, to estimate direction. Bushy neurons are specialized for resolving these minute time differences. For high-frequency sounds, the intensity difference between ears becomes the dominant cue, since the shorter wavelengths are more easily shadowed by the head. A few individuals, particularly those who are blind, use human echolocation, emitting sounds and interpreting the returning echoes to navigate their surroundings. Balance, or equilibrioception, is another function governed by the inner ear. It allows us to move upright without toppling over, though some animals, like cats, do it with notably more grace. Balance is a matter of detecting acceleration, managed by fluid called endolymph in the labyrinthine canals of the inner ear. When this system is disturbed, the result is dizziness, disorientation, and often nausea. One can induce a temporary disruption by closing the eyes and spinning. The fluid in the semicircular canals continues moving after you stop, creating a mismatch between vestibular and visual signals, hence the spinning sensation. Astronauts in orbit experience a similar conflict due to microgravity, resulting in space sickness, a reminder that our sensory systems are built for life on Earth. Touch is the first sense to develop in the womb and the last to fade before death. It is our most primal link to the world, and fittingly, the most expansive. With roughly 50 touch receptors per square centimeter and about 5 million sensory cells in total, the skin is not only the body's largest organ, it's also among the most intricate. Touch receptors are specialized by function. Mechanoreceptors detect pressure, vibration, and texture. Thermoreceptors respond to changes in temperature. Nociceptors are responsible for the perception of pain. These various receptors work in concert to create our tactile experience. Pacinian corpuscles are the largest of these receptors and specialize in sensing gross pressure and high-frequency vibration. Structurally, they resemble tiny onions with layers of connective tissue surrounding a nerve ending. When the skin is suddenly indented, this structure deforms, opening pressure-sensitive sodium ion channels in the axon membrane. Sodium floods in, generating a receptor potential. These receptors are designed to respond to quick changes. If pressure is steady, they quickly adapt and stop firing. Their sensitivity to rapid deformation suggests a role in detecting swift changes in joint position. Meissner's corpuscles are smaller and located just beneath the epidermis within the dermal papillae. They are especially concentrated in areas of high tactile acuity: fingertips, palms, soles, lips, tongue, the face, nipples, and external genitals. These receptors respond to light touch and low-frequency vibration. Being phasics, or rapidly adapting, they fire quickly when touched, then stop responding even if the stimulus remains. When the stimulus is removed and the corpuscle returns to its resting shape, another burst of action potentials is generated. This phenomenon, called H sensory adaptation, explains why we often stop noticing the feeling of clothing on our skin. However, Meissner's corpuscles do not detect pain. That job is reserved for free nerve endings, which are unencapsulated and widely distributed. Ruffini corpuscles, named after the Italian histologist Angelo Ruffini, serve primarily as thermoreceptors, though they are also slowly adapting mechanoreceptors. Located in the glabrous, hairless dermis and subcutaneous tissue, these spindle-shaped receptors respond to skin stretch. They contribute to the kinesthetic sense, helping us detect finger position and movement, a quiet but essential part of fine motor control. Together, these specialized structures give rise to a nuanced sense of touch, allowing us not only to feel, but to navigate, interact, and respond with precision to the physical world around us. The circulatory system is essential to sustaining life, tirelessly delivering oxygen and nutrients to every cell, while also removing carbon dioxide and other metabolic waste. It helps regulate pH, distributes immune cells and proteins, and supports countless other physiological processes. When this system falters, often from slow, progressive arterial deterioration, the results are serious. Heart attacks and strokes remain leading causes of death in many developed nations. While the circulatory system technically includes the heart, blood vessels, blood, lymph, and lymphatic vessels, it is often shorthand for the vascular network anchored to the heart. In practice, circulatory and cardiovascular are frequently used interchangeably, though strictly speaking, the latter refers to the system of blood and blood vessels alone. Our focus here is that network and the heart that powers it. The heart, about the size of a closed fist, begins its rhythmic contractions within weeks of conception and continues unceasingly until death. Over an average lifetime, it may beat around 3 billion times, roughly 100,000 times per day, for an 80-year-old. This relentless pump circulates blood through an estimated 60,000 miles of vessels. Blood itself carries oxygen, nutrients, hormones, and immune agents, while also transporting pathogens and waste products for disposal. Anatomically, the heart is a hollow muscular organ made of cardiac muscle, a type found nowhere else in the body. It sits in the thoracic cavity between the lungs and contains 4 chambers that function as a double pump. This muscle is self-exciting, meaning it initiates its own contractions independent of direct neural input. However, its rhythm can be influenced by the nervous and endocrine systems. For instance, physical exertion or the perception of threat can cause the heart rate to accelerate. Arteries carry blood away from the heart. This includes both oxygenated blood, such as that in the systemic arteries, and deoxygenated blood, as seen in the pulmonary arteries. Their thick 3-layered walls, composed of endothelium, smooth muscle, and connective tissue, are designed to withstand and respond to the high pressure of blood ejected from the heart. These walls are capable of expanding as blood surges through them. Arterioles, small branches of arteries, lead into the capillary networks. These too have muscular walls that can contract or dilate to regulate blood pressure and flow. The more arterioles dilate, the lower the systemic blood pressure becomes. Though just visible to the naked eye, they play an outsized role in circulatory regulation. Capillaries are the smallest vessels, forming the connection between arteries and veins. With a combined surface area of approximately 6,300 square meters, they are so widespread that no cell in the body is more than 50 micrometers away from one. Capillary walls consist of a single layer of endothelial cells, making them ideally suited for diffusion. Oxygen, water, and lipids pass out of the blood and into tissues, while waste products like carbon dioxide and urea re-enter the blood for disposal. These capillaries are arranged into networks called capillary beds, which can open and close depending on local metabolic demand. This process, called autoregulation, ensures that tissues receive blood flow in accordance with their activity level. Veins return blood to the heart. The pulmonary veins carry oxygenated blood, while systemic veins transport deoxygenated blood. At any moment, roughly 70% of the body's blood volume is in the venous system. Veins have the same three-layered structure as arteries, though with thinner walls, less smooth muscle and connective tissue, and operate under lower pressure. Because of this, veins rely on surrounding skeletal muscles to help push blood upward against gravity. Many contain one-way valves to prevent backflow, particularly in the limbs. When blood pools, such as during long periods of standing or inactivity, it can lead to varicose veins. The internal channel through which blood flows in a vein is called the lumen. Veins also possess a muscular layer that enables them to contract slightly, pushing more blood back toward the heart. They are commonly used in medicine for drawing blood, venipuncture, or for delivering treatments directly into the bloodstream, intravenous therapy. Venules, the smallest veins, collect blood from capillary beds and pass it along to larger veins. Like arteries and arterioles, they consist of 3 layers, but contain less smooth muscle and are therefore thinner and more flexible. In its entirety, the circulatory system is a vast, efficient, and indispensable network, one that, though often taken for granted, works without pause from our first heartbeat to our last. The respiratory system is indispensable to life beyond the womb. It ensures that oxygen enters the body and carbon dioxide is expelled. With each breath, whether drawn in or pushed out, a coordinated effort of cavities, tubes, and openings moves air to and from the lungs. The path of this airflow is known as the respiratory tract, which is divided into 2 sections, the upper respiratory tract and the lower respiratory tract. The upper portion includes the nostrils, nasal cavities, pharynx, epiglottis, and larynx. The lower tract comprises the trachea, bronchi, bronchioles, and lungs. Air is warmed, moistened, and filtered as it travels. This filtration begins at the nostrils, where coarse hairs trap large particles. Inside the nasal cavity, blood vessels help warm the air, and mucus-producing epithelial cells filter out finer particles. Cilia, tiny hair-like structures on these cells, sweep mucus and debris toward the pharynx, where they are either expelled or swallowed and neutralized by stomach acid. From here, the air passes through the pharynx and then to the larynx. The larynx, or voice box, lies just below the split where the pharynx divides into the trachea and esophagus. It contains the vocal cords, connective tissue folds that vibrate to produce sound, and the epiglottis, a cartilage flap that closes over the trachea during swallowing to prevent aspiration. When the epiglottis fails in its duties, choking results. From the larynx, air moves into the trachea, a tube supported by incomplete rings of hyaline cartilage that keep the airway open. These rings are open at the back, where a band of smooth muscle, the trachealis, allows the trachea to constrict if needed. The trachea is lined with ciliated, mucus-secreting epithelium that continues to filter inhaled air. It terminates at the carina, where it splits into the right and left primary bronchi. The bronchi branch repeatedly into smaller passages, secondary or lobar bronchi, then tertiary or segmental bronchi, and finally into bronchioles. As these branches narrow, the amount of cartilage decreases and smooth muscle increases. The smallest bronchioles give way to terminal bronchioles, which lead to respiratory bronchioles, alveolar ducts, and ultimately the alveoli. Alveoli are tiny hollow sacs that serve as the functional units of the lungs. Lined by type 1 pneumocytes, these simple squamous cells allow for rapid gas exchange. Type 2 pneumocytes secrete surfactant, a substance that reduces surface tension and prevents alveolar collapse. The lungs contain approximately 300 million alveoli. The lungs themselves are cone-shaped organs in the thoracic cavity. The right lung, larger and divided into 3 lobes, superior, middle, inferior, features both horizontal and oblique fissures. The left lung has 2 lobes, superior and inferior, and includes the cardiac notch, an indentation to accommodate the heart. At the medial surface of each lung is the hilum, where vessels and bronchi enter and exit. Each lung is enveloped by a visceral pleural membrane, while the thoracic cavity is lined by a parietal pleural membrane. These serous membranes secrete pleural fluid, which reduces friction and helps the lungs adhere to the chest wall during breathing. The respiratory system supports 4 key processes: ventilation, breathing; external respiration, gas exchange between lungs and blood; internal respiration, exchange between blood and tissues; and cellular respiration, the use of oxygen to produce energy in cells. Beyond oxygen delivery and carbon dioxide removal, this system helps regulate blood pH, defends against pathogens, and contributes to thermoregulation through evaporative heat loss during exhalation. The right to speak and sing, to cough and yawn, even to sigh, all depend on this delicately balanced system, from the movement of air to the vibration of vocal cords, from the cilia in the nasal passages to the surfactant in the alveoli. Without it, life would be quite literally breathless. The immune system is our body's defense infrastructure, a diligent and often underappreciated guardian against a world teeming with pathogens. It operates through 2 main strategies: non-specific and specific immunity. Non-specific immunity, also called innate immunity, doesn't discriminate. It's like a fence around your property. Everyone is kept out, whether friend or foe. This broad-spectrum defense acts the same way regardless of the intruder's identity, working to prevent entry or destroy pathogens outright. It includes mechanical barriers, chemical defenses, cellular components, and the process of inflammation. Mechanical barriers are straightforward enough. The skin and mucous membranes form a physical blockade against pathogens. Beyond just sitting there like a brick wall, they actively work to remove unwanted substances. For example, mucus in the respiratory tract moves particles toward the digestive tract, and tears flush irritants from the eyes. Not elegant, but efficient. Chemical substances contribute another layer of defense. Mucus doesn't just trap particles, it also carries them to the stomach, where powerful digestive enzymes await, eager to destroy. Among the chemical defenders are cytokines, proteins secreted by cells to combat pathogens. A notable example is the interferon, a cytokine that binds to cells and prompts them to produce antiviral substances, effectively suppressing viral replication. Some interferons even rally immune cells like macrophages and natural killer cells. Then there's interleukin I, a cytokine with a flair for drama, inducing fever in response to toxins or pathogens, a biological signal that something is amiss. The complement system is another notable player. It's a cascade of about 20 plasma proteins, designated C1 through C9, along with factors B, D, and P. Once the first in the series is activated, the rest follow suit in a domino effect. This cascade produces inflammation, attracts phagocytic white blood cells, and targets non-self cells for destruction. Quite the multitasker. Inflammation, the hallmark of local immune response, announces itself with swelling, redness, heat, and pain, tumor, rubor, calor, and dolor, respectively. It's triggered by tissue damage from trauma, temperature extremes, or chemicals. The goal here is to increase blood flow to the site, bringing repair materials and containing the spread of any would-be invaders. Histamine and heparin, released by mast cells, take the lead. Histamine dilates vessels and makes them more permeable, while heparin slows clotting. Phagocytes rush in, clean up debris, and neutrophils signal fibroblasts to begin repairs. As a side effect, certain substances also stimulate local pain receptors, lest you forget that something has gone wrong. Shifting to specific defense, also known as adaptive immunity, we move from a generalist approach to targeted strikes. This system not only identifies specific pathogens, but remembers them, launching stronger responses upon future encounters. Adaptive immunity divides into two branches: cell-mediated and antibody-mediated. In cell-mediated immunity, T lymphocytes, T cells, take center stage. These cells become activated when they encounter antigens, specifically the molecular fragments known as antigenic determinants, or epitopes. Each T cell carries receptors on its surface, composed of polypeptide chains with variable and constant regions. The variable region binds directly to the epitope, initiating a targeted immune response. Major histocompatibility complex molecules, or MHCs, also play a critical role in this activation. These glycoproteins reside on cell membranes and feature a variable region designed for antigen binding. There are two main types: MHC class I and MHC class II. MHC class I molecules present antigens produced inside cells, such as viral proteins from an infected host cell. These are displayed on the cell's surface, essentially waving a flag that says, "Something's wrong in here," prompting the immune system to destroy the infected cell outright. MHC class II molecules, in contrast, appear on cells that specialize in presenting antigens. These antigens come from outside the cell and enter via endocytosis, then pair with MHC class II molecules in vesicles. The resulting complex is shuttled to the surface for display. However, unlike the class I presentation, this isn't a call for destruction. Instead, it serves as a call to arms for other immune cells, signaling them to coordinate a response against the identified pathogen. Which organ is the most important in the body? It's a common debate, often reduced to a contest between the heart and the brain, but rarely does anyone nominate the gastrointestinal tract, even though it spans over 30 feet and plays a role so central to life that its failure can precipitate a slow unraveling of health. Indeed, in many cases, the beginning of the end can be traced not to the heart or brain, but to the intestines. The more accurate version of the old saying, "You are what you eat," might be, you are what you digest and absorb. This is the essence of the gastrointestinal system's function, digestion and absorption. Together, these processes allow the body to transform a sandwich or a salad into usable fuel, delivering vital nutrients to trillions of cells. The gastrointestinal tract is a long, continuous tube that begins at the mouth and ends at the anus, passing through the esophagus, stomach, small intestine, comprising the duodenum, jejunum, and ileum, and large intestine, which includes the colon and rectum. It's not the most aesthetically pleasing collection of organs, but its importance is profound. If the body is like a donut, then the GI tract is the donut hole, technically outside the internal environment of the body, even as it performs internal functions. Even before food enters the mouth, the digestive process can begin. The mere sight or smell of food can trigger salivation, a preparatory response from the body that signals what's to come. Once food is in the system, digestion breaks it down into basic units the body can actually use. Proteins must be dismantled into amino acids, carbohydrates into simple sugars, and fats into fatty acids and glycerol. This breakdown occurs through two concurrent processes, mechanical and chemical digestion. Mechanical digestion involves the physical breaking apart of food. It starts with mastication in the mouth and continues with the churning action in the stomach, reducing food into smaller particles in preparation for chemical breakdown. Chemical digestion, on the other hand, involves enzymes, biological catalysts that transform macromolecules into absorbable units. This process begins in the mouth with enzymes in saliva and continues throughout the intestines, where an arsenal of specialized enzymes works tirelessly. The digestive system itself includes both the alimentary canal and accessory organs. The alimentary canal is the tube itself, the mouth, esophagus, stomach, and intestines, through which food passes. Meanwhile, accessory organs like the liver and pancreas contribute crucial substances. The liver produces bile, an emulsifier that assists in fat digestion. Bile is stored and concentrated in the gallbladder during fasting, and released into the small intestine when needed. Food doesn't simply go into the body when it's swallowed. It passes through the digestive tract, propelled by smooth muscle contractions. Only when nutrients are absorbed through the intestinal wall and enter the bloodstream or lymphatic system can they be considered inside the body. In the small intestine, absorption is the key activity. Nutrients, proteins, fats, carbohydrates, vitamins, and minerals cross the intestinal lining into circulation, where they can be delivered to cells throughout the body. Meanwhile, in the large intestine, water is reabsorbed, along with some minerals. The remaining material is compacted into feces and eventually eliminated through the anus. The muscular system is the biological engine behind movement in the human body. Though not as flashy as the brain nor as rhythmically vital as the heart, it is essential all the same. In vertebrates, this system operates under the command of the nervous system, though some of its operations, such as those of cardiac muscle, can function autonomously without input from the central command center. Muscle tissue itself is contractile in nature, derived from the mesoderm during embryonic development. Its primary role is to generate force and produce motion. This motion may be external, as in moving limbs and digits, or internal, such as the pulsing of the heart, or the rhythmic squeezing of the intestines known as peristalsis. Much of this activity goes on entirely without conscious control, just as well, since we have better things to do than think about each heartbeat or the progress of lunch through the small intestine. On the other hand, voluntary muscle contraction, such as flexing a bicep or typing, is under our direct control and allows for both precise and broad movements. Muscle is made up of individual muscle cells, also called muscle fibers. Inside these cells lie myofibrils, and within those are the sarcomers, the true workhorses of muscle function. Sarcomers are composed of actin and myosin filaments, which slide past each other to create contraction. Each muscle cell is wrapped in a layer called the endomysium. These cells are bundled together into fascicles by a layer called the perimysium. The fascicles are then grouped to form the complete muscle, surrounded by the epimysium. Scattered throughout the muscle tissue are muscle spindles, sensory structures that report back to the central nervous system, providing feedback on muscle stretch and position. Skeletal muscle, perhaps the most well-known variety, is organized into distinct groups. The biceps brachii, for instance, is connected to bones by tendons and serves to move parts of the skeleton. Smooth muscle, by contrast, appears in nearly every organ. In the skin, it's responsible for making hair stand on end. In the digestive system and blood vessels, it regulates movement and diameter, respectively. There are about 640 skeletal muscles in the human body. Contrary to popular belief, exercise does not increase the number of muscle fibers. Rather, it causes existing cells to grow larger, a process known as hypertrophy. In some cases, the internal structures, myofibrils, can multiply when exposed to sustained stress. Despite their variety, all muscle types, smooth, cardiac, and skeletal, rely on the same fundamental mechanism: actin filaments sliding over myosin to generate contraction and relaxation. Skeletal muscle contractions begin with a nerve signal that triggers the release of acetylcholine at the neuromuscular junction. This chemical messenger generates an action potential along the muscle cell membrane, leading to contraction. Many smooth muscles are activated in a similar fashion. Muscular work consumes a great deal of energy, and muscle tissue stores some of this energy locally in the form of glycogen, which makes up about 1% of a muscle's mass. When energy demands increase, glycogen is rapidly broken down into glucose. As for the three muscle types, smooth muscle, also known as involuntary muscle, consists of spindle-shaped cells found in the walls of internal organs and structures, including the esophagus, stomach, intestines, bronchi, uterus, ureters, bladder, and blood vessels. Each smooth muscle cell contains a single nucleus and lacks the striations seen in skeletal muscle. Cardiac muscle is also involuntary but differs in appearance. It is striated like skeletal muscle and also has cells with a single nucleus. This type of muscle is found exclusively in the heart, where it keeps things pumping, thankfully without needing a reminder. Skeletal muscle, or voluntary muscle, is what moves the skeleton. It is attached to bones via tendons and responsible for locomotion and posture. These cells are multinucleated, with nuclei located toward the edges of the fibers, and appear striated under the microscope. Beyond moving limbs, skeletal muscle helps maintain body temperature, supports the body's structure, aids in blood and lymphatic circulation through its contractions, protects internal organs, and helps stabilize joints. Cardiac and skeletal muscles are considered striated, a term that refers to the presence of sarcomeres. organized units of actin and myosin that form a repeating pattern. These sarcomeres are bundled in highly regular arrangements, giving the tissue a striped appearance under the microscope. Smooth muscle, in contrast, lacks this organization, and thus the striations. It also tends to function differently. Striated muscles, like those in your limbs or your heart, are typically used in short, powerful bursts of activity. Smooth muscle, on the other hand, is built for endurance. It can sustain prolonged or even continuous contractions with remarkable efficiency. Skeletal muscle itself comes in a variety of forms, optimized for different types of work. It can be divided into several subtypes based on function, color, and metabolic properties. Type Y muscle fibers, often referred to as slow oxidative, slow twitch, or simply red muscle, are densely packed with capillaries and contain a high concentration of mitochondria and myoglobin. This combination gives the tissue its red appearance and allows it to support sustained aerobic activity. It's the type of muscle you rely on for endurance tasks, like maintaining posture or running a marathon, assuming you're the sort of person who runs marathons. Type 2 fibers, classified as fast twitch, come in several subcategories arranged by increasing speed of contraction. Type 2A is similar to Type 1 in that it's aerobic and rich in both mitochondria and capillaries, and it also appears red. It can sustain activity longer than other fast twitch fibers while still offering quick response times. Type 2X, sometimes labeled as Type 2D, sacrifices endurance for power. It contains fewer mitochondria and less myoglobin than the oxidative fibers, enabling faster and more forceful contractions. However, it tires quickly and is best suited for short, intense efforts. In humans, this is the fastest muscle fiber type. Curiously, it was once misidentified in some literature as Type 1IB, which led to some taxonomic confusion. True Type 2. B fibers are anaerobic, glycolytic, and appear white due to their low myoglobin content. While these fibers are rare in humans, they are common in small animals like rodents and rabbits, which explains the pale appearance of their muscle tissue. Skeletal muscle contractions are typically the result of conscious control. The brain generates an action potential that travels down the nervous system to a motor neuron, which in turn stimulates the muscle fiber to contract. However, not all muscle activity originates in the brain. Reflexes, for instance, are quick, automatic reactions to unexpected stimuli. These are coordinated through the spinal cord, bypassing the brain entirely for the sake of speed. Muscle contractions occur in three broad categories, each corresponding to the type of muscle involved. Skeletal muscle contractions, responsible for voluntary movement. Cardiac muscle contractions, which drive the heartbeat. And smooth muscle contractions, which keep things like digestion, blood flow, and other involuntary processes going, often in the background, and mercifully without conscious effort. The skeletal system plays a rather foundational role in the human body, not just literally in terms of support and movement, but also in storage and production. It is a reservoir for calcium and other inorganic salts, and a bustling factory for blood cell formation. Comprising 206 bones of assorted shapes and sizes, the adult human skeleton is a complex and multifunctional structure. Bones come in four basic shapes. Long bones, such as the femur, feature a prominent longitudinal axis. Short bones, more cube-like, are compact and efficient. Flat bones are thin and often curved, like those found in the skull. Then there are irregular bones, which defy categorization, often found in clusters with unique varied forms. Examining a typical long bone reveals a few standard components. The diaphysis, the central shaft, is composed of dense compact bone and encloses the medullary cavity, which houses bone marrow. On either end of the bone is an epiphysis, formed of cancellous or spongy bone. The epiphysis is covered with hyaline cartilage, facilitating articulation with neighboring bones. Between diaphysis and epiphysis lies the epiphyseal line, the vestige of the once active growth plate. Encasing the entire bone is the periosteum, a fibrous membrane rich in blood vessels and cellular machinery vital for bone growth and repair. Bone tissue itself comes in two forms: compact or cortical bone and cancellous or spongy bone. Compact bone is structurally dense and forms the outer shell of most bones. Cancellous bone, with its trabecular or lattice-like matrix, is found primarily in the epiphyses of long bones and within flat bones. Despite its airy appearance, cancellous bone is metabolically active and essential for functions like hematopoiesis, the formation of blood cells. Compact bone is arranged into structural units, known as haversian systems or osteons. These are aligned with the direction of force, giving bones their impressive strength. Each osteon includes a central haversian canal that serves as a passageway for blood vessels and nerves. Around this canal, bone is deposited in concentric layers called lamellae. Within these layers are lacunae, tiny spaces that house osteocytes, the mature bone cells. Canaliculi extend from each lacuna, forming a network through which nutrients and signals travel. The entire system is further interconnected by larger passageways known as Volkmann's canals, or perforating canals, linking one osteon to another. Bone is maintained and remodeled by three main types of cells. Osteoblasts, which arise through mitosis, secrete osteoid, a soft matrix that later hardens with mineral deposits. These cells respond to hormones and physical stress, adjusting bone growth accordingly. Once trapped within the matrix, they helped build. Osteoblasts become osteocytes, which no longer divide but maintain the bone tissue. Osteocytes reside in the lacunae. In contrast, osteoclasts are the demolition crew. They demineralize bone, releasing calcium into the bloodstream when needed elsewhere. Within the medullary cavity of long bones, and in the spaces of some spongy bones, lies bone marrow. In infants and children, red marrow dominates, producing red blood cells in abundance. With age, much of this red marrow is replaced by yellow marrow, composed largely of fat cells and less proficient at blood cell production. Nevertheless, several adult bones retain active red marrow, including the proximal ends of the humerus and femur, the ribs, vertebral bodies, and the pelvis. Structurally, the skeleton is divided into two main regions. The axial skeleton includes the skull, spine, rib cage, and sacrum, essentially the central core. The appendicular skeleton comprises the limbs and girdles, attaching to and extending from the axial framework. Together, these systems provide both stability and mobility, protection and productivity. The integumentary system is composed of the skin, hair, nails, subcutaneous tissue, and various glands. Its most visible role is protection. The skin acts as a barrier against harmful substances and helps retain bodily fluids, performing this function quietly and continuously. Beneath the skin, the subcutaneous tissue connects the dermis to deeper tissues such as muscle, creating a stable, integrated structure. Hair plays its own small but useful part. On the scalp, it offers insulation. Eyelashes and eyebrows keep dust and sweat from entering the eyes, and nasal hairs trap airborne particles before they reach the respiratory tract. Nails protect the tips of the fingers and toes from mechanical damage, and with their firm backing, assist in handling small objects. There are 4 types of exocrine glands in this system: sudoriferous, sebaceous, ceruminous, and mammary glands. Sudoriferous glands, responsible for sweat, help regulate body temperature. Sebaceous glands produce oil that inhibits bacterial growth, waterproofs the skin, and prevents drying. Ceruminous glands produce earwax, which maintains ear canal pliability and protects the eardrum. Mammary glands, found in the breast, produce milk. Skin itself is a complex, multitasking organ. It shields muscles and organs, provides thermal insulation, facilitates sensation, and even synthesizes vitamins D and B. It also serves as the first line of defense against pathogens. Melanin, produced by melanocytes, absorbs ultraviolet radiation and offers some protection from its damaging effects. These cells also contain enzymes that repair UV-induced DNA damage. Without those enzymes, susceptibility to skin cancer rises sharply, especially to forms like malignant melanoma, which can spread rapidly and prove deadly. Human skin pigmentation varies considerably across populations, a variation that has, unfortunately, been the basis for misguided classifications. When skin is damaged, it repairs itself through scar formation, which may result in discoloration or loss of pigmentation. The skin, often referred to as the largest organ in the human body, lives up to the title both in surface area, about 1.5 to 2.0 square meters in the average adult, and in weight, comprising roughly 15% of body mass. A single square inch holds a dense collection of features, about 650 sweat glands, 20 blood vessels, 60,000 melanocytes, and over 1,000 nerve endings. Across cultures, it's common to use both natural and synthetic cosmetics to modify or maintain the appearance of the skin, particularly of the face. Pore control and blackhead cleansing are perhaps not biologically essential, but they do seem to hold widespread appeal. Structurally, the skin has 2 main layers, the epidermis and the dermis. Below these lies the hypodermis, or subcutaneous adipose layer, which, while not technically part of the skin, plays a supporting role. The epidermis consists of stratified squamous keratinizing epithelium. It lacks blood vessels, and instead receives nourishment by diffusion from the underlying dermis. It is primarily composed of keratinocytes, though melanocytes and Langerhans cells are also present. The epidermis is arranged in strata from outermost to innermost: stratum corneum, lucidum, granulosum, spinosum, and basal. New cells form through mitosis in the basal layers, migrate upward, change shape and composition, and eventually become part of the outer corneum before being sloughed off, a process known as keratinization, which takes about 30 days. This layer is vital for retaining body water and keeping harmful agents out. Capillaries run beneath the dermis, branching off from arterioles and feeding into venules. In certain areas, arterial shunt vessels can bypass these networks, particularly in the nose, ears, and fingertips. The dermis, located beneath the epidermis, is where much of the functional machinery resides: blood vessels, nerve endings, hair follicles, smooth muscles, lymphatic vessels, and glands. It's made of loose areolar connective tissue with collagen, elastin, and reticular fibers lending it strength and flexibility. Attached to each hair follicle is an erector muscle that contracts under stress or cold, producing the familiar phenomenon of goosebumps. The primary cell types here include fibroblasts, which produce connective tissue, adipocytes, which store fat, and macrophages, which defend against pathogens. Sebaceous glands secrete sebum, a lipid-rich substance that moisturizes, waterproofs, and protects the skin with antibacterial properties. Sweat glands open onto the surface via pores, playing a central role in thermoregulation. The dermis can be divided into two layers. The papillary layer, the upper portion, extends into the epidermis and supplies it with nutrients. It contains loosely arranged fibers and forms the ridges on our fingers and palms, better known as fingerprints. The deeper reticular layer is more robust, composed of irregularly arranged fibers, and anchors much of the skin's structural components. It connects seamlessly with the underlying hypodermis and resists stretching, ensuring the skin stays in place, even under stress. The hypodermis, although not technically part of the skin, lies just beneath the dermis and serves several structural and functional roles. It anchors the skin to underlying bones and muscles and supplies it with essential blood vessels and nerves. Composed of loose connective tissue and elastin, its dominant cell types include fibroblasts, macrophages, and adipocytes. The hypodermis holds about 95% of the body's fat, providing insulation and cushioning, soft armor of sorts. The skin itself performs numerous functions vital to survival. First and foremost, it offers protection, acting as an anatomical barrier between the internal and external environments. Immune cells like Langerhans cells help to fend off microbial invaders. It also serves as a highly sensitive sensory surface, detecting heat, cold, pressure, vibration, and injury. In terms of thermoregulation, the skin has a generous blood supply, allowing for nuanced control of heat loss through vasodilation and vasoconstriction. In animals, the contraction of tiny erector pili muscles serves to trap heat by raising hairs, though in humans, the effect is mostly aesthetic, resulting in goosebumps. Human hair comes in three distinct types. Lanugo is the fine, unpigmented hair covering a fetus, mostly replaced by birth. Vellus hair, also unpigmented and soft, is the body's general coating, especially in children and adult females. Terminal hair, by contrast, is longer, thicker, and pigmented, appearing in areas like the scalp, face, especially in males, armpits, and pubic region. Fingernails made of keratin perform two key functions. They protect the fingertips and enhance tactile sensation. With their high concentration of nerve endings, fingertips are sensitive tools. The nail acts as a counterforce, increasing the pressure when an object is touched, and thus heightening sensory feedback. Each nail is composed of 6 defined parts. The rootie, or germinal matrix, lies beneath the skin and is responsible for most nail production. It lacks melanocytes, which is why the lunula, the white crescent-shaped area at the base of the nail, is pale. The nail bed, or sterile matrix, supports the nail plate and contributes to its thickness. It contains nerves, blood vessels, and melanin-producing melanocytes. If this surface is damaged, nail growth can become distorted. The nail plate is the visible part of the nail. It's composed of translucent keratin and gets its pinkish hue from the underlying vascular nail bed. Grooves beneath the plate help secure it in place. The eponychium, or cuticle, seals the space between the skin and nail plate, forming a waterproof barrier. The cyperioecium refers to the skin on the sides of the nail plate and is the common site of hangnails and infections like paronychia. Finally, the hyponychium lies under the free edge of the nail, providing another barrier between the external environment and the fingertip. Sweat glands fall into two categories, each with different functions and outputs. Eccrine glands are found all over the body, but are especially concentrated on the palms, soles, and forehead. These glands are responsible for regulating body temperature by producing sweat, primarily water, with a mix of salts and waste products. Structurally, they're coiled tubular glands that originate in the dermis and empty directly onto the surface of the skin. Eccrine glands are activated by sympathetic cholinergic nerves, which are themselves regulated by the hypothalamus, a structure that monitors core temperature and receives input from skin sensors. Depending on internal and external cues, the hypothalamus adjusts sweat production accordingly. The composition of human eccrine sweat includes small amounts of fatty acids, urea, and other waste products. Sodium concentrations range from 35 to 65 millimoles per liter and tend to be lower in people acclimated to hot climates. The sweat of other species can vary considerably in content, particularly among marine and fur-bearing animals, many of which lack eccrine glands altogether. Apocrine sweat glands develop around puberty, typically by age 15, and initially produce excessive amounts of sweat for a short time before settling into a more regulated pattern. These glands are concentrated in specific regions of the body, particularly the armpits and the area surrounding the genitals. Unlike eccrine glands, apocrine glands produce sweat that contains fatty substances. While the secretion itself is odorless, its breakdown by skin bacteria results in the familiar and often unwelcome scent of body odor. Apocrine glands are activated not by heat, but by emotional stress. Their sweat is already present in the ducts and is released in response to stress signals. In function, they behave more like scent glands than cooling mechanisms. Sebaceous glands are present throughout the skin of mammals, with the notable exception of the palms and soles. These glands secrete an oily substance known as sebum, a mixture of lipids and the remains of disintegrated fat-producing cells. In most areas of the body, sebaceous glands are attached to hair follicles, delivering sebum to the surface via the hair shaft. This trio of hair, follicle, and gland is collectively referred to as a pilosebaceous unit. Sebaceous glands are also found in hairless regions, such as the lips, eyelids, genitalia, and nipples, where they release sebum through ducts directly onto the skin. Sebum's primary functions are to lubricate and waterproof the skin and hair, preventing dryness, cracking, and microbial invasion. Though odorless when freshly secreted, it can develop an odor when broken down by skin bacteria. The result of accumulated sebum can be seen in oily hair left unwashed for several days, and in the crusty residue found in the corner of the eyes after sleep. In addition to its role in general skin maintenance, sebum contributes to the composition of earwax, along with other secretions. The biochemical makeup of human sebum is roughly 41% triglycerides, 25% wax monoesters, 16% free fatty acids, and 12% squalene. Sebaceous gland activity is stimulated by androgens, which is why oil production typically increases during puberty. This can also explain the acne that tends to flourish during adolescence. Excess sebum, along with clogged pores, often leads to skin disorders like acne and keratosis pilaris. blocked glands can form sebaceous cysts, and conditions like acne may be treated with isotretinoin, a medication that dramatically reduces sebum output. Conversely, excessive use of anabolic steroids, common among certain bodybuilding circles, can stimulate sebaceous activity and exacerbate acne. In utero, the sebaceous glands of the fetus secrete a substance called vernix caseosa, a waxy white coating that protects the skin of the developing newborn. In some species, sebaceous glands have taken on specialized roles. The preputial glands of mice and rats, for instance, are large, modified sebaceous glands that secrete pheromones. Ceruminous glands are another modified structure, specialized glands in the ear canal that produce earwax, or cerumen. This yellowish, waxy material serves several protective functions. It helps trap dust and foreign particles, maintains moisture, and wards off bacteria, fungi, and even insects. Cerumen is formed in the outer third of the ear canal and is made from a combination of thicker secretions from sebaceous glands and lighter ones from modified apocrine glands. While beneficial in moderation, excess cerumen can accumulate and press against the eardrum or block the ear canal, leading to impaired hearing. Earwax, or cerumen, comes in two genetically distinct types, wet and dry. The wet type, moist and ranging in color from honey brown to dark brown, is dominant and more commonly found among individuals of African and Caucasian descent. The dry type, which is gray and flaky, is recessive and predominantly found in people of East Asian and Native American ancestry. Interestingly, anthropologists have used cerumen type to trace ancient human migratory patterns, including those of the Inuit. This difference in earwax type has been traced to a single nucleotide polymorphism in the ATP binding cassette C11 gene. Beyond affecting cerumen composition, this genetic variation also reduces sweat production, a change thought to be advantageous for the ancestors of East Asians and Native Americans who likely inhabited colder climates. Cleaning of the ear canal is a self-sustaining process. It relies on the conveyor belt migration of epithelial cells, beginning at the tympanic membrane center and moving outward to the ear canal's entrance. The rate of this migration is comparable to fingernail growth. As cells move outward, they carry cerumen and any debris it has collected toward the external opening. Jaw movement helps dislodge particles stuck to the canal walls, enhancing this natural self-cleaning process. Lubrication provided by cerumen is critical. It prevents the drying and itching of skin within the ear canal, a condition known as osteotosis. This lubricating quality is due to the high lipid content of sebum, which in wet-type cerumen includes cholesterol, squalene, and long-chain fatty acids and alcohols. Cerumen also has antibacterial and antifungal properties. Although early research was inconclusive, more recent studies show that cerumen is effective in reducing the viability of various bacteria, including Haemophilus influenza, Staphylococcus aureus, and several Escherichia coli strains, sometimes by as much as 99%. It also inhibits the growth of fungi associated with automycosis. These protective effects are largely due to saturated fatty acids, the enzyme lysozyme, and cerumen's mildly acidic pH, typically around 6.1. Mammary glands are specialized organs found in female mammals for the production of milk. These are enlarged and modified sweat glands, fitting, since they're part of the integumentary system, and are the very feature from which mammals take their name. Each mammary gland is composed of alveoli, small hollow structures lined with milk-secreting epithelial cells. These are surrounded by contractile myoepithelial cells. Alveoli group into lobules, each of which drains into a lactiferous duct that opens at the nipple. When myoepithelial cells contract, they push milk through the ducts into small reservoirs known as sinuses, which collect the milk until it is expelled, typically by the pressure of a suckling infant. A simple mammary gland includes all milk-producing tissue draining into a single duct, while a complex mammary gland encompasses all simple glands serving a single nipple. Humans typically have two complex glands, one in each breast, each composed of 10 to 20 simple glands. Occasionally, individuals have extra nipples, polythelia, or extra complex glands, polymastia, both of which are benign anomalies. Mammary gland development is hormonally driven. Though present in both sexes, these glands remain rudimentary until puberty. Estrogen stimulates development in females, whereas testosterone suppresses it in males. At birth, infants have only lactiferous ducts. True development of alveoli and branching ducts occurs at puberty under the influence of ovarian hormones. During pregnancy, estrogen and progesterone cause further alveolar development, an increase in adipose tissue, and enhanced blood flow to the region. In late pregnancy and shortly after birth, the glands secrete colostrum, a nutrient-rich pre-milk fluid. Full lactation begins a few days postpartum as progesterone levels fall and prolactin takes over. The act of nursing stimulates the release of oxytocin, which causes the myoepithelial cells to contract, effectively squeezing milk out to meet its eager recipient.